Today I will be talking about mitosis and meiosis. Now both mitosis and meiosis are forms of cellular reproduction. Cellular reproduction is important in repairing damaged tissue, like whenever you get a cut, growth and development, like how you get a little bigger every year, and reproduction, like having a baby. To start off, we will be talking about mitosis. When it comes to mitotic cell division, the cell divides to produce two cells called daughter cells. Those daughter cells are genetically identical to the mother cell, so they are an exact copy. To do this, the cell has to go through a few steps. To remember these steps, prophase, prometaphase, metaphase, anaphase, and telophase, you can remember the acronym PPMAT, or like a puppy PMAT, like this one here. However, before any of those phases, there's interphase. During interphase, the DNA replicates itself so there is now double the amount of DNA in the nucleus than there was before. In prophase, the chromatin begins to condense to distinct chromosomes. The centrosomes, which are these, start to migrate to different sides of the cell, and mitotic spindles, which are the little things coming out of it, start to push the centrosomes to opposite sides. During prometaphase, the sister chromatid pairs are fully condensed. The nucleus that you previously saw is now breaking down, so the mitotic spindles can start to connect to the sister chromatids at their connector cores. There are other microtubules that don't connect with the sister chromatids, but interact with each other so that later on they can help elongate the cell. In metaphase, the sister chromatids line up at the metaphase plate, which is dead center of the cell. The sister chromatids have microtubules connected to both sides of them so that there is an even pull from both sides. During anaphase, the protein that normally keeps the sister chromatids attached is cut so that mitotic spindles can pull them apart to their opposite sides. The microtubules that interact with the other microtubules begin to help to elongate the cell. In telophase and cytokinesis, the nucleus will start to reform and the chromosomes will begin to unravel. During cytokinesis, which happens around the same time, the cytoplasm is eventually split between the two cells. The cell is then pinched by the cleavage furrow to separate them into two identical daughter cells. Now, before any cell is able to go through mitosis, they have to pass through some different checkpoints. The cell kind of asks itself, am I big enough to divide? Did I replicate all of my DNA? Is the DNA damaged? Are my chromosomes aligned properly at metaphase? Checkpoints can also be activated via external stimuli like growth factors. However, if these checkpoints are ignored in mitosis, it can cause many problems like different kinds of cancers. Now we can talk about meiosis. Meiosis is another form of reproduction. It can be done asexually or sexually. Asexual reproduction means that the offspring inherits genes from a single parent, whereas sexual reproduction means that the offspring inherits genes from two parents. Now you might ask yourself, how do sexually reproducing organisms keep the same number of chromosomes from generation to generation? Well, chromosomes are passed to offsprings by the fusion of gametes. The female gamete is the egg, whereas the male gamete is the sperm. Gametes are haploid cells. Now, normally, human body cells are diploid, like the cells in your liver or the cells in your brain. These diploid cells have a total of 64 total chromosomes. However, gametes are haploid, which means that they are half the number of cells. So, the haploid cells only have 23 chromosomes. So during the cycle, you'll get half your chromosomes from mom and half your chromosomes from dad, which come together to make a diploid zygote having 46 chromosomes. Now, the steps for meiosis are pretty simple. The DNA replicates before, so originally there were 42 or 46. Then you multiply that by 2 and you get 92. So this number for this original cell is 92. It then goes through similar step process that was in mitosis, where it splits into two different cells, but now both of these have 46. It doesn't replicate again though, but it goes from this meiosis 1, which is this where it first separates into two, and then goes into a mitosis 2, where each of these separates into two also. So now this 46 separates again, so now there's 23 and 23. And this 46 also separates to 23 and 23. So in meiosis, you start with one cell, but you end up with 4 daughter cells, instead of in mitosis, where you start with one cell, but you end up with 2 daughter cells. Now, during the meiosis cycle, it'll ask itself similar checkpoint questions to that of the mitosis cycle, like, am I big enough to divide, or is any of my DNA damaged? But the most important question is, are all of my chromosomes aligned at the metaphase plate? If this is ignored, something called non-disjunction can happen, which is a failure of normal separation. This could either mean that too many chromosomes or too few chromosomes are within the cell. Example where it's bad to have too many chromosomes is something that we all know called trisomy 21, which is more commonly known as Down syndrome. In a normal karyotype, 21 only has 2 sets of chromosomes, whereas in trisomy 21 for Down syndrome, there are 3 sets of chromosomes. Thank you all for watching this video. I hope you learned a little something about mitosis and meiosis, and I'll see you next time. Bye!